What you cuties doing today? Are you guys watching Andy Griffith's head taped to a TV? Well, this episode is a standalone, technically, but it would be also very fitting as a sequel. Transcription 2, a trip to protein-topia. As it starts right where we left off in the last video, with RNAP releasing RNA strands to be decoded. Firstly, we should cover the term codon, which refers to a short sequence of only 3 nucleotides. A codon can sequence for either their corresponding amino acids or to signal the initiation or termination of translation processes. Secondly, there is the workhorse of this process, the totes bodacious ribosome, an absolute unit with grooves for fittin' and slots for sittin'. Ribosomes also can catalyze to form linkages between the amino acids being brought by tRNA. Speaking of, there are 3 different types of RNA we're going to focus on. tRNA, rRNA, and mRNA. That's spelled with an M. I said M as in Mancy. Each plays a role in turning specific sequences into their corresponding proteins, but must work together to do so. mRNA, the direct product created by RNAP during transcription. Once the process of splicing, where introns are removed from the hot-off-the-press mRNA strand, is finished, it's ready for the ribosome to process. mRNA acts as the template the ribosome will use tRNA to build proper proteins with bases on its specific codons. Which brings us to tRNA, my favorite of the RNAs, really. This determines which segment of code corresponds with which amino acid. This little loop on the tRNA contains the anticodon. It wields 3 nucleotides that fit to a codon on the mRNA. Sitting at the opposite end is the corresponding amino acid, rRNA. This stuff is a bit funky, as this RNA isn't itself coded, but is still very important for coding nonetheless. rRNA is the most abundant type of RNA found within cells, and is used as a primary structural component for ribosomes. That's right, cuties, rRNA not only makes up a large chunk of ribosomes, but it is what drives protein synthesis between tRNA and mRNA. Now that the actors and actresses are introduced and the stage is set, let's get on with this funky little dance. The ribosome's major and minor portions gently hug the awaiting mRNA, where it is read 1 genetic codon at a time, and the corresponding amino acid is linked to the growing peptide change. Big props to tRNA for this step. The start codon is always AUG, and is the key that kicks off the whole process. The next step is elongation, where codons continue to match with respective tRNA molecules and the amino acid chain continues to grow. During this process, codons are shifted and exposed in sequential order to be matched and snatched through the A, P, and E slots of the ribosome. Termination is the last major step, and it begins where the start codon's counterpart, the stop codon, enters the ribosome, letting it know to separate the different components to let the polypeptide chain float away to fulfill some mysterious higher purpose elsewhere in the body. Alright, you punks, I hope you learned yourself a learn. This'll come in handy once the zombie uprising cometh in 2046 on March 31st at 2:03 p.m.